Welcome to this session on the chemistry of organic chemistry in biology and drug development. We are now in the we are covering the second part of this course, and we are already through with the general concepts of drug design principles and followed by high throughput screening and combinatorial chemistry, basically how to arrive at a heat very quickly, and then we started discussing about different kinds of drugs. Basically these are case studies. So, we have started with the drugs acting on the neurotransmission, that means the chemistry of neurotransmitters, followed by the chemistry of antimicrobial agents, and then we discuss the antiviral compounds. And now today we will be discussing the chemistry and biology of a disease which is yet to be conquered, and that is what is known as cancer. Okay. Now, some few words before we go on to the actual chemistry, that how can we design drugs which are aimed towards eradicating cancer. Cancer still remains one of the most feared diseases in the modern world. Okay. It is known for a long time, but the actual cure or the proper cure is still not in our hand. According to the World Health Organization, it affected 1 person in 3, that means 1 in 3 person is going to die, and that was the statistics in 2000. So, after that almost 20 years have gone by. So, cancer is on the rise. It is not that it is on the downward trend. Basically, heart disease is the one which is the major cause of death, followed by cancer. Okay. Now, the problem of cancer, that what is the problem with treating cancer is, one problem is that the migration of the cells from one place to another. That means, this is a process which is called as metastasis. That means, that usually what happens, that the cells which are forming my liver or my heart, they remain at that position. Liver cells will never migrate to other position, or heart cells will not migrate. But in cancer what happens, there is a growth somewhere, and that growth is so uncontrollable that it finally migrates from one place through the bloodstream to any other organ or tissue. So, that is the major problem, and that is what is called the malignancy. So, okay. There are other types of now this growth is what is called tumor, that we know, and if the tumor stays there, that there is no metastasis, that means that is what is called a benign tumor. Okay. So, a tumor becomes malignant when it has the capacity from migration of one place to another. Okay. And now the question is, that is one major problem of cancer, but the question is, why can't we get a cure for that? And the problem lies with the fact that the cancer cells belong to the host itself. Okay. It is not that it is caused by some organism coming from outside. What is that what happens in case of antibacterial bacterial infection or viral infection or fungal infection. So, you can use different chemicals to kill those, and there is a distinction between bacterial cell life and versus the host. So, we can exploit that what is the differences that exist. The problem with cancer is that because it belongs to the host itself. Okay. So, while the malignant cells, we are talking about malignant cells, the malignant cells have some minor differences with the normal cells, but there are some differences, and that is the one which have been exploited or tried to be exploited whatever the cancer development, drug development has progressed. Okay. So, what I am saying that because it belongs to the host, so there is only minor differences or there may be some differences which are yet to be discovered. That is, of course, we must mention that there must be some differences due to which some cells become cancerous, that means they have uncontrollable growth, and but whatever differences are known today, so people have or scientists have exploited that. Okay. So, another point is that microorganisms we kill by antibacterial agents, that is true, but ultimately what happens that even if we take antibiotics, then ultimately few cells of the bacterial origin will remain in my body, and now the remaining cells basically are taken care of by the immune system in our body. In case of viral infection, it is the immunity which is very important to eradicate the virus, the viral infection, but in cancer somehow this immunity does not work. Okay. So, the cancer cells evade the immunity. Now, one explanation is that it belongs to the host. Immunity is always developed against foreign organism. Okay. However, if some cells become what are called rogue cells, that means the cells which are not behaving properly, still the immune system they can take care of that, but however, cancer somehow evades that immune system. Again I repeat what I am saying, that immune system usually works against foreign organisms, but even in the case of host, if some cells become unruly, the rogue cells, okay, there are in that case also immune defense can work, but in case of cancer, the rogue cells, the cancerous cells, they somehow evade the immunity. However, some recent progresses have been made towards that aspect, because if one has to develop a holistic approach to treat cancer, then possibly it is the immune system ultimately that we have to bank on, because immune system is a very general strategy, whereas the cancers could be in different varieties, different forms, because it can be head and neck cancer, it could be cancer of the liver, it could be cancer of the pancreas. Each cancer has their own differences, okay, their own identity. So, the same drug cannot work for all the types of cancer. Some drugs work for breast cancer, some drugs work for prostate cancer, but if you want to have a holistic approach, then possibly it is the immunity which has to be understood properly and then exploit that to kill the cancer. So, today basically what we will do, we will first discuss that what are the targets for developing anti-cancer drugs. We know targets are the most important, that is the initial, that is the first aspect of drug discovery program, that you want to know the target. It could be a protein, it could be nucleic acid, or it could be a small molecule. All these are targets. Now, after identifying the targets, then we will discuss that different drugs against the identified targets, and then discuss their chemistry. Chemistry means basically discuss their mechanism of action. How do they work? Okay. Until I know that, we cannot come out with a new molecule. Okay. The new molecules are based on the existing molecules, existing drugs. Okay. So, let us start that we told about I mentioned that cancer cells have an uncontrollable growth, whereas the normal cells have a definite life cycle pattern. Okay. It does not grow uncontrollably, the normal cells. When it is required, it will grow. When it is not required, either it goes into a phase which is called a resting phase, where it does not divide, or if the cell becomes bad for some reason, that if the DNA is not proper, if it is broken, sometimes what happens, they are marked to be destroyed, okay, and that is what is called programmed cell death, or also called apoptosis. Okay. So, apoptosis is basically death of a cell, but this is a programmed death. Not all cells are apoptotic. Okay. Some cells, when the body decides that this cell cannot do the job, cannot do the function properly, first it tries to repair that, but if it is not repairable, it is usually programmed to death. Okay. On the other hand, if the signal comes that I do not need any growth any further right now, so the cells go into what is called a G0 phase or a resting phase. Okay. But if there is this mitosis, that means when the cell division takes place. So, before cell division, all these important molecules like DNA has to be replicated, and then the different proteins have to be expressed. So, all these have to be done before the mitosis takes place. Okay. So, it all starts with this G1 phase, and then G1 phase, and then it goes to the S phase. This is G1 means the gap 1 phase, it is called gap 1. That means, it is now preparing the cell for synthesis of the DNA. Okay. Synthesis of the DNA means the replication of the DNA, that is required. So, gap phase 1, and then after the synthesis is done, then there is again a gap phase 2. So, it prepares the cell for the mitosis, for the cell division. Okay. So, the first one prepares the cell for the synthesis, and the second one prepare the cells for mitosis. Okay. And after the mitosis means now everything is done. So, they divide, and one copy lives with the parent cell and the one with the daughter cell. Okay. Let us see, I think I have a yes. Now, there are safety points like the checkpoints. Means if something is processed, like in the making of a car, see, you have to make all the parts and then assemble them, and finally put the tires and everything, and it is ready to go. Okay. Similarly, in case of cells, you have to make all the parts, but the question is whether all the parts have been made properly or not. So, there are checkpoints. Okay. And checkpoints means there are proteins which will see that everything is working, or everything has been assembled right in a right fashion. Okay. So, these are the proteins like CDK, they are called CD kinase. They are cyclines, they are collectively called cyclines, but they are basically kinase enzymes. Kinase is basically what does the phosphorylation of the of say serine weight or threonine weight. These are the common phosphorylation sites. Okay. Now, these cyclines actually do the check that everything is here, cell is well prepared. So, then it push the cell towards the synthesis phase, and after the synthesis is done, then there are other cyclines like cyclin A. It checks whether the DNA has been has the correct information, the copy has been correct or not. Okay. If it is not correct, then it will now throw. That is what I am saying, that it will try to correct it. If it can correct, then it is okay, otherwise it will push it into the apoptotic cycle, and then once everything is checked, everything is correct, then you have this mitosis, that means the cell division takes place. Okay. In cancer, somehow this process, this programmed process does not take place, cancer cells. Okay. Somehow it just goes on and on, that means it always goes via the G1 phase, S phase, G2 phase, and the mitosis, the M phase. Okay. All the time it is growing. So, that is what is basically it is a breakdown of the program of cell cycle. That is what causes, what basically causes cancer. But this is cause means this is the, we are talking about the cycle, that life cycle, that why it grows all the time. But what causes this life cycle to break down, that is the major issue, because that is the major point between the difference of a cancer cell and the normal cell. I think whatever I said is, it is written here. Control of cell cycle involves a variety of proteins called cyclins and enzymes called cyclin dependent kinases. Cyclin and cyclin dependent kinases, so basically both are proteins, but when they are bound with each other, then it can show the phosphorylation power. Okay. There are 15 types of cyclin, 9 types of CDK. Each has a role to play at different stages of cell cycle. Binding of a cyclin with associated kinase, that what I said, that when it binds, the enzyme activates the enzyme. Enzyme is the kinase enzyme. Okay. The protein is the cyclins, and this enzyme cyclin complex then checks the basically serves to move the cell from one phase to the cell cycle to another. For example, when a cell is in the G1 phase, a decision has to be made whether to move to the S phase and start copying the DNA. This decision is taken depending on the balance of stimulatory versus inhibitory signals being received through signal transduction. Okay. So, that is another point. The point is that when a cell goes from G1 to S phase, then that decision has to be taken. Now, the decision depends on that whether there is a need for growth or there is a need for inhibition of growth. Okay. So, it depends on the various signal processing and ultimately the balance of that. Balance means what is the overall direction, that whether to move forward or stop there. Okay. So, that is basically what it is saying, depending on the balance of stimulatory, that means which says that you have to grow, or the inhibitory signals which says that do not grow. So, it is a balance between the two, and if the balance is towards cell growth, then cell division will take place and a particular type cyclin D will be increased. Okay. This binds to the kinase 4, CDK4 and CDK6. Okay. So, that is basically it, that the importance of this kinases, and there are drugs. That means, now you can say that if I can find a drug which binds to the kinase and stops the, or binds to cyclin and do not allow the binding of the cyclin dependent kinase, then that will be that can be a good way of stopping the growth of the cancer cell. Okay. We will come back to that. Okay. I think there are some points still there. The resulting complex, that means the cyclin and cyclin dependent kinase, phosphorylate a powerful growth inhibitory molecule known as PRB, which normally binds and inactivates a transcription factor. Transcription factor is the one, you remember that when the transcription factors are one which actually starts the process of the transcription. Okay. They are called transcription factors. That means, from where the transcription has to take place, there are some promoter sites where the transcription factor binds, and then the system knows that the DNA has to be copied. Okay. So, these transcription factors, what happens, that when these cyclin, the CDK, that means the kinase and the cyclins, okay, they form a complex that phosphorylates a powerful growth inhibitory molecule PRB, okay, which normally binds and inactivates a transcription factor. Okay. Try to understand cyclin CDK complex, and then there is a powerful growth inhibitory factor which is called PRB, that is a protein, a transcription factor. So, that becomes phosphorylated. So, phosphorylated PRB. This phosphorylated PRP binds and inactivates the transcription factor, which is necessary for the transcription. That means the growth will be inhibited if the PRB is phosphorylated. However, phosphorylation alters PRB such that it no longer bind to the transcription factor. Sorry, it is the other way around. The resulting complexes phosphorylated a powerful growth factor which normally binds and inactivates a transcription factor. Okay, the switch is making a problem. I think we will start from fresh. একটু বলে দাও তো আবার প্রথম থেকে করতে হবে গো। ব্যাপারটা কি এখানে ইংরেজিটা এমনভাবে রয়েছে যার ফলে গন্ডগোল হয়ে গেল। ও যেটা বলতে চাইছে ফসফোরিলেশন করলে আবার প্রথম থেকে একটু, কারণ এই জায়গাটা একটু গন্ডগোল হয়েছে। একটু দ্বারা আমি বলে দিচ্ছি। মলিকুল known as PRB which normally binds and inactivates, which normally binds, আর ফসফোরিলেশন নয়, ফসফোরিলেশন চেঞ্জ করছে ট্রান্সক্রিপশন ফ্যাক্টরটাতে এবং ফ্রি টু বাইন্ড স্পেসিফিক রিজিয়নস alters PRB such that it can no longer bind to the transcription factor, and the latter is free to bind specific regions of DNA, so that it can no longer না ঠিকই বলছিলাম বোধহয় দেখি হ্যাঁ হ্যাঁ ঠিকই আছে রে দাঁড়া দাঁড়া ওকে ডাকি ঠিকই আছে ফসফোরিলেট করলে এবার গ্রোথ ফ্যাক্টরটা ঠিকই আছে তাহলে আমার ঠিক জায়গা থেকে বলবি আমাকে সিগন্যাল দিবি ঠিক আছে আপনি কোথা থেকে এই ফিল্ডটা দিয়ে স্টার্ট করবেন কি এই তুমি একটু দেখে আসবে দেখো তো একটু দেখে না দেখছি আমি সেটা বলে দিচ্ছি ও যাচ্ছে Okay, we have already told that these two important proteins that play a role in pushing the cell from one phase to the other, and the protein what is called the cyclins and the enzyme like protein is called the kinase, that is CDK. Okay. Each cyclin has a counterpart kinase, okay, CD kinase. Okay, now what happens, the cyclin and kinase that forms a complex, and this complex binds to another protein which is called PRB, and so the PRB becomes becomes phosphorylated. So it becomes phosphorylated. Okay, phosphorylated. Now when it is phosphorylated, then the PRB protein, actually the PRB protein, its function is that PRB protein binds to what is called a transcription factor. Transcription factor are the ones which will which activates the genes basically. There are promoter regions, so it activates the genes. So PRB usually binds to the transcription factor, and in that bound form transcription factor is not active. Okay. So it cannot promote the gene expression of the gene. So what happens after the phosphorylation is done, if the PRB is phosphorylated, then what happens, it cannot now bind, it falls off from the transcription factor. So the transcription factor is now free to bind to the promoter region, and once it binds to the promoter region, so there will be activation of the genes, and then that will ultimately lead to production of proteins, and these proteins are capable of moving the cell towards the S phase. So we are talking about the cell going from the G1 to G2 phase. So in that case PRB protein is very important. Phosphorylation means it cannot bind to the transcription factor, and dephosphorylation means now it is bound to the transcription factor. So that is what is the nature's way of controlling the growth of a cell. This is very important. So phosphorylation and dephosphorylation. Phosphorylation, again I repeat, phosphorylation means the transcription factor is free, so the cell will now go from the G1 to the G2 phase. Similarly, there are other checkpoints like when the G2 to S phase, then S phase to the G1 to S phase and S phase to the G2 phase, that is also will be checked, and basically G1 and G2 are the ones which are called preparatory stages, okay, where these balance and checks are made, and then the cell is pushed forward for the M phase, that is the mitotic phase. Okay. So, what I was telling that these kinases, now you can tell that these kinases are important targets for discovery of anti-cancer agents, because that is a way to control the growth of a cell. Okay. To sum up, the progression through the cell cycle is regulated by sequential activation of cyclins and CDKs, a process which can be down regulated by the CDK inhibitors. What I said, that its down regulation means that in the downstream processing, that when the cell moves further for the mitotic towards the mitotic stage, that is called downstream processing. So, in the down, it will be down regulated by the CDK inhibitors. Okay. The whole process is normally tightly controlled, such that there is an accumulation of relevant cyclin CDK complex, followed by rapid degradation of the complex once its task is complete. It is so tightly controlled that this complex will be formed, phosphorylation will be done, and then once the job is done, then again the complex will be broken, and if the next cycle again the complex has to be formed, it will form the complex. Okay. Overactive cyclins means with several cancers. Overactive cyclins or CDKs, kinases, means that the cell is growing very fast. Okay. For example, breast cancer cells often produce excess of cyclins D and E, and skin melanoma, another type of cancer of the skin, has lost the gene that codes for the inhibitory protein. There are some inhibitory proteins which are named as P16 or P53. All these are very important. This P16 is the gene which codes for the it is a gene which codes for the protein P16. Okay. That is an inhibitory protein. So, skin melanoma, when you study, you see that okay, P16 is gone, it is not there. In case of breast cancer, you see excess of cyclins. Do not be too means just say that cyclins are more overexpressed in case of breast cancer. In skin melanoma, it is the other way around, the inhibitory proteins are missing. That means the corresponding gene is missing. Half of the human tumors lack a proper functioning of P53 protein, which means that the level of inhibitory protein P21 falls. There are many inhibitory proteins. P16 inhibitory means which inhibits the growth. P16, P53 is the original one, and then P21. So, these proteins actually control the growth. Actually, they have an inhibitory effect on the growth. So, what has been found that there is a lack of proper functioning of P53 protein. There is this also the level of P21, and then as I said in skin melanoma, P16 protein is lost. Okay. So, that is and in some of there are different types of cancer. I told you there are these viral related cancers. Both PRB and P53 proteins are often disabled. Okay. The virus disables these proteins which safeguards our body. Okay. So, nature has our body has a natural defense mechanism via these P16, P53, P21, okay, and then PRB. But they are if they are malfunctioning, then the problem is that will be growing very fast, and also if there is excess cyclins. Cyclin activity more means more growth. Okay. So, you have some idea that what could be the if you consider the cell cycle, that what could be the targets, that cyclins could be a target, the kinase could be a target, or somehow you have to see why P53 protein is produced is down regulated. So, you have to see all these, and then that is some differences with the normal cell. So, you can design molecules which will activate or deactivate some of these. Okay. Now, let us come to the what are the actual the drugs that we have, what do they target. Okay. Now, as I said, in the S phase means the synthesis part, that is the synthesis. Now, for the before the mitosis your mitotic stage, what happens? You have to prepare, you have to make all the molecules which are needed for a cell to perform its activity. Okay. Now, it all starts with what? It all starts with the that first you have to the body has to get, or the cell has to get this pyrimidine and the purine bases. So, they have to be synthesized, biosynthesized, okay. And then you assemble sugar plus this pyridine or purine bases, and then what you get is the ribonucleotides, okay, coming from ribose. Then ribonucleotide is reduced to what is called deoxyribonucleotide, because you know that the 2' wedge is missing in the DNA, but that actually comes from the ribonucleotides. And ribonucleotides, then the deoxyribonucleotides, basically they form the double stranded DNA, and from the DNA you get the messenger RNA, and finally the messenger RNA gives the proteins which are enzymes, and also they also produce another proteins. These are called microtubules, which are extremely necessary to segregate the cell into daughter cells, okay, that mitotic process. And so, basically what you need for the cell to complete the S phase and then go to the M phase, to the G2 phase, see, you can stop any of these, or even if a cell is in the resting phase, you can target the cell by targeting any of these processes. Okay. So, everything is a target, like if you target the formation of the ribonucleotide. Okay. So, you have a drug, because that cannot proceed further. You can target this enzyme which is called ribonucleotide reductase, because then the deoxyribonucleotides will not be produced. You can directly target the DNA, so that the process of transcription does not take place or replication does not take place. And then you can target the messenger RNA also, that is possible, like I gave you some example that the messenger RNA cannot, the flow of information cannot take place if you use what are called antisense compounds, that which binds to a portion of the RNA, okay, by use of the antisense RNA, and then the translation process will be stopped. So, you can stop the translation process, and then at the enzyme level you can also target different enzymes which are vital for the growth of the cell, okay. And also microtubules, because this is the one which does the cell division, okay. So, these are the different targets, and on both sides you have different kinds of drugs, okay. I am not naming those drugs, but we will take few of these, that how do they really work, okay. Now, out of these all these targets, you can say that the best target to destroy a cell is the nucleic acids, the DNA, because DNA is the basically the commander in chief of this cell cycle, whatever is going on, all the cell processes. So, if you kill the commander in chief, then because all information is coming from the commander, so the whole battalion will fall, okay. So, that is exactly what happens. The major target of anti-cancer drug development is the nucleic acids, and followed by other different processes. I will tell you that I will just take not all, because this is itself is a lecture by itself, is not a lecture but a course by itself, the development of anti-cancer drugs, but we will try to finish up as quickly as possible, just taking some of these as the key examples like methotrexate. Okay, we have already done the methotrexate. Methotrexate, what it does, it inhibits the dihydrofolate reductase, which is important for making the for the thymine from uracil. Okay, so that is one of the earliest anti-cancer drug that was developed. Now let us go one by one. I think so. First, we will go by the DNA as the target, nucleic acid as the target. Okay. Now, nucleic acid, how many ways you can target the nucleic acids? It says by alkylating agents. What are alkylating agents? Alkylating agents are nothing but like if you have a phenol, if you add methyl iodide, the phenol will form OAB. If you have a nitrogen amine, and if you add say again methyl iodide or dimethyl sulfate or even diazomethane, that will form the alkylation, the N alkylation. Now, there are different alkylation, N alkylation, C alkylation, O alkylation, S alkylation, all these are possible. So, alkylating agents, what they do in DNA, actually the most nucleophilic part is the bases. We have nitrogen bases, and there are the nucleophiles that are present, and out of these nucleophiles, it is if you probe the structure of all these bases, you will see that guanine has a very strong nucleophilic nitrogen in the form of that imidazole part, because guanine is imidazole fused pyrimidine, and that imidazole nitrogen which is by the way called N7 nitrogen, that is the most nucleophilic part. So, what happens if you have a alkylating agent, not a single alkylating, which has got the ability to make suppose X, and it has got alkylating agent, means you should have a good leaving group, a nucleophile comes, attacks, and this leaves. Okay. So, if you have a bis alkylating agent, so one nucleophile comes from here, another nucleophile comes from here, and both leaves. So, what you will have? You will have a bis alkylation. Okay. So, if you have this DNA which is double stranded, and if you can do a bis alkylation, so what will happen to the DNA? The DNA will now see. Again, there are different types of cross linking. You have this DNA like this. So, what I am saying that suppose there is a G here, the G is cross linked, and then there is a G here, that is also cross linked. So, that means this cross linking is what? This cross linking is between two strands, this strand and the other strand, there is a cross linking. So, basically after cross linking it will become something like this. So, that will be bent at that point, but the most important point is that as there is a cross link, so these two strands cannot be separated there. You cannot separate at that point, because there is a knot there. The other type of alkylation that can happen is that the same strand, that is called, this is called inter-strand cross linking. Inter, you can have intra also. Intra means the same strand, like here there is an alkylation site and there is an alkylation site here. So, you now join these two. So, the whole thing becomes a kind of a oval shape at that point, and that also, see, the fallout of this is basically your replication and transcription stops, because you have a deformed DNA in this case, and in this case there is a knot which you cannot release, that is not possible. Okay. So, that is alkylating agent. Chain cutters, that is even more direct, that you have an agent which breaks the double strand DNA into two pieces. So, I just showed in this form, in this cartoon picture, this is the double stranded DNA, you have a scissor, these chain cutters are also called artificial scissors. Okay. Intercalating agents. Intercalating agents means DNA is double stranded and you have these bases, and there are certain planar aromatic compounds which can go inside. That is lot of space between these base pairs, and then another flat aromatic ring can go inside and then sit there and stabilize the DNA. That is what is called intercalation. So, if there is intercalation, then also it will require lot of forces first of all to separate, because another stabilizing force is there, okay, as the intercalated molecule, and also there will be change of shape, because to accommodate that intercalating agent, the DNA has to has to change a little bit to accommodate that partner. Now, you can say why it is accommodating it, because overall it gets a stabilization, because it is called pi-pi stacking. See, the base pairs have a pi cloud, and then you have a planar aromatic kind of system, and then another base pair. So, there is always pi-pi stacking that stabilizes the DNA. So, these are the three different types of DNA stopping the DNA from transcription. Okay. But there is another way of stopping the transcription, is topoisomerase inhibition. Topoisomerase, you know that when the DNA unwinds, there is this supercoiling in front. So, topoisomerase takes care of that supercoiling, but if you can inhibit the topoisomerase, then also your transcription will stop. Okay. So, these are the 4 classes of agents that will target the basically the transcription process, the replication and the transcription process. The first, one of the drug which goes via intercalation is what is called doxorubicin. Doxorubicin is, you see, doxorubicin has this aromatic ring. It is an anthraquinone based compound called anthracyclines, because not only anthraquinone, you have a cyclic part here. These are called anthracyclines, and they are planar. So, they go inside. This is the molecule which goes inside the double stranded DNA. So, this is dox DNA, that means it is already the doxorubicin is inside. So, when it is inside, you it cannot replicate or undergo transcription properly. So, this is doxorubicin is one which is, it is trade name is adriamycin. It is a chemotherapeutic medicine to treat cancer. It binds to DNA through intercalation. So, what is the mechanism? Through intercalation. So, basically weak forces that play a part here, no covalent forces. Okay. It is used for many cancers, breast cancer, bladder cancer, Kaposi sarcoma, this is a kind of skin cancer, lymphoma, then acute, so many, leukemia. Okay. However, always remember, I told you there are different types of cancer, So, only one single reagent cannot cure the cancer. Okay. So, you have to give a cocktail of different anti-cancer drugs. So, whenever doxorubicin is given, at the same time other anti-cancer drugs are also given, which work on a different principle, so that the cells do not become resistant later on, because here also the cancer cells become resistant to the drug which is used earlier, which has been used earlier, and by a mechanism which is the efflux mechanism. I told you that some of the bacterial cells also have that efflux mechanism, that it pumps out the entering drug. So, these cancer cells also have very efficient efflux mechanisms, and then can remove the drug very quickly, so that it cannot enter the cell. Okay. Topoisomerase inhibition, I told you that if you can inhibit the topoisomerase, then you can also get anti-cancer action, and this is a very important natural product called camptothecin. Camptothecin is the one which actually here it says topoisomerase inhibition in bracket poisons. Actually, what happens here, inhibition is in topoisomerase. Remember what I said at that time during your transcription, that there is that supercoiling ahead of the replication fork. So, to bypass the supercoiling, to reduce the supercoiling, what the topoisomerase does, it forms a nick at some point, and then it turn it over the other strand, so that the supercoiling effect is not felt. And once your replication fork proceeds, then it again comes back and closes that whatever nick it has produced earlier. Okay. Now, what happens in this case, topoisomerase forms the nick, and then this nick is basically stabilized, that DNA part with the nick is stabilized by the topoisomerase. Now, what happens, this camptothecin, what it does, it usually that topoisomerase and the nick DNA complex is very transitory. Transitory means it has to be transitory, because as the replication fork moves, it has to again join it back. So, but the camptothecin molecule or these type of topoisomerase inhibition molecules, they what they do, they stabilize that whatever was transient earlier, they stabilize that nick DNA topoisomerase complex. So, that is why it is usually sometimes called poison. It is a topoisomerase poison. Truly it is not an inhibition, actually the topoisomerase first do the first does the job of nicking, forms a transient complex, and the transient complex again is broken and the nick is again repaired. But here that repairing process cannot take place, because the transient complex is stabilized. Okay. So, that is camptothecin, and there are different analogues of camptothecin people have made, and those are also used as drugs. Okay. You just see the type of heterocyclic framework that camptothecin has. It has, this is called a quinoline, and this is an isoindole. So, that is the part, but the thing is that this is also quite planar in that sense. So, it can go and somehow stabilize the DNA topoisomerase complex, nick DNA topoisomerase complex. Okay. Now, let us come to the alkylating agents. It also says metallating agents, because alkylation also can be done by, it is not alkylation, sometimes what happens, you can have a metal ion and that is stabilized by, because these whatever are nucleophiles can be good ligating agents also to metals. Okay. So, this is also possible. A metal ion is bridged between the two bases of different strands, and that also forms what is called double metallation. Okay. Double metallation is nothing but double alkylation. Okay. So, now the alkylating agent, as I told you, this is the nitrogen which is most susceptible to the, this is N7, most susceptible to the your attack N alkylation, and also the cytosine this nitrogen N3, because of this nitrogen lone pair, and here because of this nitrogen lone pair. So, that increases the nucleophilicity. So, let us see what are the structures of these alkylating agents. Now, there is a history behind discovery of this alkylating agent. There is a compound which are called sulfur mustard, because sulfur mustards look like the, it is yellowish in color, look like a mustard. Mustard also has yellow color. Okay. So, this is the compound called sulfur mustard, and sulfur mustard is very interesting that it is a double alkylating agent, and the alkylation proceeds via what is called neighboring group participation, that the sulfur first attacks the carbon, the chlorine is lost, and that forms what is called an episulfide, like your epoxide. So, it is an episulfide, and then you have this CH2Cl. So, this becomes plus. Now, the suppose the G with the nitrogen comes here, attacks there, and the sulfur opens up, and the process is repeated again. Another G comes here, and then via the neighboring group participation it becomes. So, this belongs to the, either could be the same strand or it could be belong to the different strand. Okay. Now, this sulfur mustard is a highly cytotoxic agent and vesicant. Vesicant means which causes blister, and it was used in World War I as well as World War II, and also in recently in the Gulf also this has been used in Syria and parts of Iraq. Okay. So, this compound basically at that time it was in World War I and II, what was happened that this was used as a chemical warfare agent. But what they found, that the persons who have been exposed to the sulfur mustard, so when they analyzed their blood count, what they found, that they have they develop what is called a leukemia. Okay. And why leukopenia is low white blood cell count, and with a bone marrow aplasia, defective development. Okay. and ulceration of the, so lot of biological effects. Again I repeat, low white, actually it is a low white blood cell count, that is sulfur mustard, and then bone marrow aplasia, which means defective development. That means it has got some effect on the growth of the cell, because defective development means it has some effect on the growth. Now, all these lesions means whatever the inflammation that we are talking about, like ulceration, defective development, all these things have a profound effect on rapidly dividing cells, suggesting that these compounds might be effective as anti-tumor agents. So, this is basically a direct fallout of the World War I, and then mustards came into the market after the World War II, okay, because people realized that apart from this chemical warfare agent, apart from producing the blisters, all these things, but it actually also it works by rapidly dividing cells. It stops the rapidly dividing cells from dividing. That means, what are the rapidly dividing cells? It is the cancer cells. So, it will have effect on the cancer cells, and however, the sulfur mustard is extremely unstable. So, what they resorted to is what is called nitrogen mustard. So, they nitrogen mustard, they are related to sulfur containing mustard gases, which was used in World War I, that I already said. Nitrogen mustard give you some ways to manipulate the structure, because remember sulfur is bivalent. Okay. So, you cannot attach anything on the sulfur except the two alkylating arms. But if you have a nitrogen, then apart from these two the chloroethyl handles, which are alkylating agents, you have a NH here. So, you can put a R group here, and that R group can be can give you the stability. Okay. Here the mechanism is that instead of the episulfide, you will get aziridinium ion as the transiently formed reactive species. Okay. But here also this ankymeric assistance is taking place. Now, depending on the R, you have different alkylating agents. The first alkylating agent which is used as a drug was this chloromethane. Chloromethane is nothing but you have a methyl here, okay, and that is the mechanism. The guanine is attacking here, and then another guanine is attacking the other aziridinium ion. So, the result is that you have a intermolecular cross link. This is intermolecular. Remember, this is the, or check, this is this for this strand, and the other alkylation is for this strand. So, that is intermolecular strand. You can also have, as I said, you can have intermolecular also, like this. Guanine from the same strands are reacting with that nitrogen mustard. So, basically this will stop the transcription process. Now, however, these molecules were very toxic in the sense that they are so small molecules that they can react with other protein molecules inside the body before it is entering a cancer cell. Okay. So, they are not the very good anti-cancer agents because of their toxicity. They can destroy the cell very rapidly, but they are not very specific in that sense. Okay. So, ultimately research ultimately on these alkylating agents gave rise to this drug cyclophosphamide. Okay. Cyclophosphamide, if you look at the molecule here, the nitrogen lone pair is now delocalized with the phosphorus oxygen double bond. So, the nitrogen lone pair cannot form the aziridinium ion, because the nitrogen lone pair is locked, because it is towards the phosphorus oxygen atom. Okay. That is a more electron withdrawing group. So, how can that means it is not a by itself, it is not a drug. So, you have to activate it. Activate means ultimately you have to break this nitrogen phosphorus bond. Okay. How was it possible that what happens when it goes to the through the liver, there is enzyme that the oxidation takes place, and specifically hydroxylation. P450 is a monooxygenase enzyme. Monooxygenase, you know, where it delivers one oxygen atom into the molecule, that means does hydroxylation. So, hydroxylation takes place at this carbon, and then this is nothing but an aminal. Aminal means like acetal, but here one of the oxygen is replaced by a nitrogen. This is also not stable. So, this becomes an aldehyde, the amine is free, and once it becomes aldehyde, the hydrogen is lost here. So, you get acraldehyde, and what you get is this compound. Okay. Still the nitrogen is locked with the phosphate. After that, there is a hydrolysis. This is spontaneous. By water, you can hydrolyze this molecule, water comes and then breaks this. So, basically it releases the nitrogen mustard in the actual form, where the nitrogen is in each. So, now it can alkylate the DNA, and it can monoalkylate, because sometimes what happens when it becomes too reactive, water can also displace the chlorine. So, there could be monoalkylation, there could be bisalkylation also, but this is the way. In monoalkylation also it is difficult for the DNA to replicate, because the DNA will be miscopied. They may not realize that this is a guanine. Okay. So, there may be miscopying possible. So, this is the mechanism. So, that means cyclophosphamide is what is called a prodrug. Prodrug means it is not the actual drug, but in the biological system it is activated, and the actual drug is nothing but this compound, and that is released. So, that is all about the nitrogen mustard. If you want to know the mechanism also, other thing that can happen, I should tell you, when you add the alkylating agent, the guanine is specifically is more preferentially alkylated, okay. And as a result there will be bisalkylation, because it is a bisalkylating agent, and there can be intermolecular or intramolecular bisalkylation, but both causes the stoppage of the replication and the transcription process. Another thing that can happen, that the DNA can lose the base when there is this type of alkylation takes place. Their DNA may be there, but in one of the DNA the base may not be there. So, it is basically only ribose, deoxyribose and the phosphate, no base is attached at the site. This is what is called abasic site. Abasic site means the base is not there. That means there is depurination, or what you say that the guanine has left. How does it leave? It is basically, you know, the what is called Maxim Gilbert sequencing method, that exactly what happens there, that you have an alkylating agent dimethyl sulfate followed by hydrolysis. Okay. So, that takes care of first the base goes out, and then the phosphodiester bond breaks. Okay. But that breakage is carried out by, if you remember, that is carried out by piperidine. Here the base is not present, means piperidine is not present. So, what happens after this after the alkylation N plus, water comes and attacks this carbon, and which ultimately results in the hydrolysis of the results in removal of the base. Because see, if you look at the mechanism, first there is plus charge, water comes, so that the electron goes here, then if this is an aminal, so this will become an aldehyde, and that will become a nitrogen NH, and then NH opens up the sugar, and this is an imine, and imine is hydrolyzed. So, basically the base goes out. Okay. So, deep that one of the base can come out, but usually it is the guanine that comes out. So, what will happen to that? For when it is copied, if the base is missing, so the enzyme, that is the polymerase, can now interpret that it could be any base. So, it can produce some different base which is not required in the mRNA. So, that also cause mutation. Okay. I think that is all about this alkylating agents. Now, they have made different types of stable alkylating agents, which are quite interesting, that you can start with aziridine, because these are not aziridinium ion. If it is aziridinium ion, that will be more reactive. So, you can have a simple molecule like this. This is a trialkylating agent. Here the driving force is that if the nucleophile attacks here, that goes there, and because of this imine nitrogen, the bond breakage becomes very fast. So, it is a trialkylating agent, this is a bisalkylating agent. Here the driving force is that the nucleophile attacks here, that goes there, that can go there, and ultimately that becomes O minus. Okay. So, different types of molecules are possible, all are working by alkylation, and but these are more stable than what we have said earlier. Cyclophosphamide is quite stable because the nitrogen lone pair has been locked in a prodrug form to start with. Okay. So, that is all about the bisalkylating agents. In the next session, we will talk about the other kinds of drugs. Thank you.